Hi everyone, we're gonna go ahead and get started. I will finish lecture 4 today, and then I'm gonna go talk about the lab. All right, so I'm gonna draw this huge chart for you as a note for the last part of lecture 4. So please take out a piece of paper, and we're gonna do that. So the title of the note is membrane transport. Okay. If you have a piece of paper, you should turn it sideways, and you should write transport here. Okay, and then we're gonna do the flow chart that I showed you a moment ago. Okay, so here we go. We're gonna have transport across cell membrane. Okay, and we have two types. So let's go all the way over here. We're gonna put passive transport here. For passive transport, we have no need for energy requirement. Okay, so no ATP required. It's going down concentration gradient. Okay, meaning it's going from high concentration to low concentration. That is what passive transport means. Now, there are different kinds of passive transport, some of which we already talked about last time, but let's do a quick summary of what they are. Okay, so for passive transport, we have simple diffusion. Simple diffusion. So simple diffusion is just the movement of substances, of molecules down its concentration gradient without any help. Okay. It is ideal for small, such as like H2O, that's small, right? Non-polar, such as, here I'll put e.g. H2O, such as O2, CO2. So these are non-polar molecules. They have even distribution of electrons. And hydrophobic. such as steroid molecules. Okay. Let me know if you guys can see what I'm writing. Like if you need me to make it bigger or if you need me to slow down, just let me know. I'll post this up online after class, of course. So we have a second type of passive transport. It is osmosis, right? Osmosis, which is basically the diffusion of water. Okay. So think about the isotonic, hypertonic, and hypotonic situation that we talked about last time, right? I remember we had quite a discussion on that, with the basin and the water coming in and leaking. Those are examples of osmosis, the movement of water across the cell membrane. Okay. I'll give you a moment to catch up. Write down what you need to. Let me know when you're good to go. Okay. Hopefully I'm not going too fast. And if I am, just let me know. Okay, the last type of passive transport, again, no energy is required, is facilitated. Facilitated diffusion. Okay. So this is to move large and or charged molecules across the cell membrane. Okay, they are either too big to cross on their own, or if they're charged, they won't be able to pass through the phospholipid bilayer, right? So we're gonna need some help. Okay, so move large and or charged molecules across the cell membrane with the help of a membrane protein. We learned about membrane proteins last time. They can be integral protein that goes through the whole cell membrane. They could be peripheral protein that just kind of attach on the outside or on the inside. And they do things like attachment to cytoskeleton, cell recognition, cell signaling, and also transport, right? And this is an example where you would need it to move molecules across the cell membrane. Now, there are two kinds of facilitated diffusion. Okay, the first type is channel protein. Channel protein. The other kind is carrier protein. Okay, they are both facilitated diffusion in that they both involve using proteins to move things across the cell membrane. But depending on how they achieve this, we call it either channel protein or carrier protein. So for channel protein, the integral protein, we'll say an integral, which means this thing goes right through the cell membrane, creates a tunnel for charged molecules to move through. Okay. The tunnel shields the charged molecule from the hydrophobic tails of the cell membrane. Okay. I can draw this out for you. Right? You probably are very familiar with this drawing by now. This is the phospholipid bilayer, and you have all these hydrophobic tails that are sandwiched in between, right? So if you have a charged particle, they won't be able to go through, which is why we are going to need a channel protein. Okay? So channel protein is an integral protein, and it has a tunnel, a channel in the middle. And then if you have all these charged particles that wants to go through, they will be able to do it like that. Okay. So I'll let you copy that down first, and then I'll explain a little bit more. I hope this works a little bit better for some of you instead of doing the PowerPoint. Sometimes it's nice to just write some notes, right? Probably will help you remember it a little bit better. I hope. Any questions? Can the page be zoomed out a little bit? Yeah, but it might get too small, right? Like I don't know. Like I assume you want to see like other stuff, right? Like that. Is that too small for some of you? Some of you on the phone, right? I believe. Yeah, I'll post this up after class. So if you miss anything, you can certainly copy after. Okay, I'm gonna move on. And I'm gonna go back in a little bit. It's easier for me to write. So the other kind is carrier protein. Right? So carrier protein is similar to channel protein. It is also an integral protein. That means, again, it goes through the entire membrane. So an integral protein changes its shape slightly to move large molecules across the cell membrane. All right. I forgot to give an example here. So it could be any charged ion, like calcium or something. Okay, and over here, large molecules would be like glucose. Okay, so they're so big that they cannot cross on their own, so they need some help. That's what facilitated means, right? If you facilitate someone, you are helping someone. So these diffusion requires help from membrane protein. That's why it's called facilitated diffusion. No energy is required. Right? So here we have a carrier protein. And let's say we have a big molecule here. Right? So it's gonna go into the membrane protein. The membrane protein will change the shape and in turns causes the molecule to be moved across the cell membrane and it's gonna go into the cell. Okay. It's like you know Pac-Man, right? Like the game where they eat the little dots, the circle, the beans, whatever, right? It just moves along. So the carrier protein will grab onto the object, change the shape, and then spit it out on the other side. Okay. Channel protein, no shape changing at all. It just sits there and let things pass through. Over here, there's a slight change in the shape. Okay. So there is an important point here that I want to mention about these two things. Okay. The protein, as in the membrane protein, does not determine the direction of movements. Okay. The concentration gradient does. Okay. So whether these charged particles are going to go into the cell or out of the cell, whether these glucose are going to go in or out, is not up to the protein. It's up to the concentration gradient. So if it's high on the outside and low on the inside, then they will come in. If the condition flipped, if it's high concentration on the inside and low on the outside, they will leave. All right, copy what you need. I forgot my water. I'm just gonna grab it. I'll be right back. Is everybody okay? Any question about passive transport before I move on to the other type? Hmm? Everybody good? Okay, let's move on to the other type. So we're gonna go over here. Okay, so for you, those of you who are planning ahead for space, we're gonna have active transport here, and then we're gonna have one type here, we're gonna have another type here, and then we're gonna have another type here and another type here, and then we're gonna have 3 types here. All right, so just plan your space accordingly. Okay, over here, all the way, we have active transport. Okay, so active transport requires ATP. And that's because you are going against concentration gradient. Meaning you are going from low concentration to high concentration. You're going uphill. Okay? So let's take a look at the first type here. First type is protein pump. All right. A protein pump uses an integral protein. Again, so an integral protein uses ATP to pump molecules against their concentration gradient. Okay. One example I can give you is called the sodium potassium pump. So here is a drawing of the sodium potassium pump. This is the inside of the cell. This is the outside. And you just have to trust me for now. I will learn this in next semester in greater details. But in order for your neurons to function properly, you need to have high sodium on the outside and low sodium on the inside. And you also need low potassium on the outside and high potassium on the inside. Okay. So how do you maintain that? Well, the sodium potassium pump is going to use ATP. Okay. For every ATP molecule that it uses, it will be bringing in 2 potassium. And then it will be bumping out 3 sodium. Okay. So if you look at the sodium, you're going from low concentration to high concentration. Right? Against its concentration gradient. Same thing for the potassium. You're going from low to high. You're going against its concentration gradient, which is why you need to have ATP to provide the energy for transporting these things. Okay. So that's just one type of protein pump. There are many different kinds of protein pump. Bacteria, sometimes they have antibiotic pumps. If you try to kill them with antibiotics, the pump will pump the drug out and it will not affect them. Right? So they are really hard to kill, right? In the stomach, we have proton pump. Right? Proton pump. That makes the acid in the stomach. If people have heartburn, severe heartburn, like taking the Tums not helping, then the doctor might prescribe you with PPI, right, which is proton pump inhibitor. There are these little pills, black and magenta, pink. If you take them, you have to take them continuously, right, to inhibit the proton pumps. so that you are not going to make as much acid. Okay. So different types of protein pumps, but that's one of them. Okay. I guess it's so quiet today. I hope you are with me. Let's do another one here. We have bulk transport. Okay. Bulk transport is to move very big molecules or large quantity of molecules across the cell membrane. Okay, so either they are really big, even transport protein can move them across, then you need to do bulk transport, or you just need to take in a lot of them at the same time. Okay. So the bulk transport can go one of two ways. You can be sending things out, which we call exocytosis. Okay. Secrete large molecules out of the cell. Right. Now maybe watch how I draw the picture first, and then you can draw it with me. So this is the cell membrane. This is the inside of the cell. This is the outside. Let's say I have a vesicle. Okay. This is a vesicle from, where do you think the vesicle is from? Do you remember from lecture 3? Where do we get these nice round vesicle that has been sorted, modified, and repackaged? Where is it from?
Isn't that like the Golgi one? It's
Body,
free
that's right.
floating.
Yep,
It's free floating in the cytoplasm.
that's right. So that's a vesicle from the Golgi body, right? Very good. Now you're right, it could be free floating, it could become a membrane protein, it could become a lysosome, but one of the paths is that it could, travel towards the cell membrane, okay, and it will get really, really close to it. It's gonna touch it. Let me see if I can touch this thing. There we go. Okay. Okay, and then it will fuse with it. It will fuse with it. Okay. I hope you can follow my series of drawing. Okay. So the vesicle basically moves to the cell membrane, It'll fuse with it, and then the content comes out, right? Okay, so an example of this would be, you know, proteins from vesicle. Okay? They get excreted. That's exocytosis, okay? Getting things out. Now, what's the opposite of exo? Exo. What's the prefix that's opposite to exo? No?
N?
Yep. You're probably right, maybe. Endocytosis. That's right, RJ. Endocytosis. Now, there are 3 kinds of endocytosis depending on what you're bringing in. Okay, but let's write the definition for endocytosis first. Instead of secrete large molecules out of the cell, we are bring in large molecules into the cell. All right. So we have 3 types. Depending on what and how you are bringing it in. The first type is phagocytosis. Okay. Phago means eating. Cyto means cell. So phagocytosis means cell eating. It brings in solid. All right. An example of that would be your white blood cells. So here is a white blood cell. It's part of your immune system. WBC is white blood cell. It sees a bacterium. Okay. And what happens is it will surround it with its membrane. And eat it up. That bacterium is not going anywhere. That's called phagocytosis. Now, nobody could tell me this morning. Hopefully, you guys are a little bit better. What organelle will come and break down the bacteria now? Which organelle is responsible for breaking down a bacterium that has just been eaten up by the cell? That's right, lysosome. Very good. Lysosome has enzymes in it that will break down the bacteria, right? If you did not know that, you should review it because your quiz is on Tuesday. Okay, next. Other than eating, what else do they do? Well, you can probably guess they drink. So penocytosis. Right? This is cell drinking. Which means they brings in liquid mostly. Okay, some extracellular fluid. Okay. An example of that would be like the human egg does that. Some kidney cells, they do that. You learn all these next semester, okay? And then there's one last type, and we're done. This is receptor mediated. endocytosis. Okay. So only molecules that match a receptor can be brought into the cell. Okay, not everything can come in. There is a little bit of selectivity here. Okay, so let's say this is the cell membrane again. And on the membrane, we have some special receptors. Okay. So you might have some red circles. Okay, you might have some blue triangles. Okay. Which one can come in? The red circles or blue triangle? Which one will fit the receptor?
The red circles?
Yeah,
The circles.
the red circle, that's right. So these fit the receptor. They can come in. Okay. That's all. There's some kind of selectivity to it. Okay. It's like going into a exclusive venue. Okay. You gotta have the right ID before they let you in. That's all. Congratulations. That's all the different types of transport. Okay. So, you know, write this out again when you are organizing your note, right? Stick it on your wall. You might find it helpful during the quiz. Okay, quiz is next Tuesday. It will be on all of lecture 3 and 4. I'll post the details later on. Okay, but I already told you the 2 short answer questions. There will be some 4 questions where you have to identify the organelle. I'm going to show you a picture of it, and you have to tell me what the name is.
Sir, sorry.
Yeah.
Can you tell us the short answer questions again?
Oh,
Which-
yeah, sure. So one of them is like in lecture 4, like something like this, right? Okay. But it might be cold environment, it might be hot environment, depending on which quiz you get. So make sure you know the answer for both. And you can watch the recording for that if you need to. And then the other question is the relationship between mitochondria and chloroplast. Okay. Relationship between mitochondria and chloroplast. Are we okay? When you label the organelle, please use the full name. Okay, don't put like RER. Okay, put like rough endoplasmic reticulum. Make sure you spell it right.
Excuse
go
me,
ahead.
sir. Can you please explain this question again?
Okay, so like we actually don't have time to explain this again, but you can watch the recording, right? For do you know where to find the recordings?
No.
Huh? No?
No.
Oh, okay. So for those of you who also don't know, if you go to East Centennial, right? Do you guys read my announcement? I post an announcement after every class.
Yes, we do. Yes.
Over here, right? It says lecture archive has been updated. You just click on that. And these are all the recordings. Okay. So probably I think it was this one that towards the end of lecture 3, we did a little bit of lecture 4. I think the explanation is here. Is it okay?
Yeah, thank you, sir.
Okay, no problem. And I will give you guys 1 hour this time. All right. Last time I gave you 45 minutes, and some of you are saying it was not enough
Thank
time.
you.
Yeah. So this time I give you 60 minutes. Okay. Should be enough.
Thank you.
Yeah. You're welcome. All right. Before I talk about the lab, I want to take up some of these questions from the PowerPoint slides. If you don't have the PowerPoint slides, you can write down the page, the slide number, and copy the answer, and then go revisit them later. You might have noticed that some of these questions show up on the quiz, right? Like when you do the first quiz last time, a few questions, at least a few questions, were taken from the concept checking stuff directly. Okay, so please review these as part of your studying process. Okay, so this is slide, what was it, 46, I think? Let me just double check. Yep, slide 46. Okay. We don't have a lot of time. I want to finish quickly, not quickly, like I want to finish early so you can start working on your lab. All right. I went all the way to like 2 o'clock basically for my morning class. So hopefully we're not going to take that long. We'll see. Passive membrane transport processes include consumption of ATP. Nope, we don't need ATP. Movement of a substance down its concentration gradient. That's the correct one. Okay. Use transport protein when moving substance from low to high. That's not true. Movement of water from an area of low concentration to an area of high concentration. That's osmosis specifically, right? This is looking for passive transport in general. So the answer is B. Slide 46. Slide 47. When a cell is placed in a hypertonic solution, what happens? So again, if you're making study notes for your quiz, make sure you have the hypertonic, hypotonic stuff, right? So net movement of water out of the cell. Okay, so you have to know what hypertonic is, right? Hypertonic is when you have a cell and then the concentration is higher outside, right, than it is inside. So water will be leaving. That's the first thing you have to remember. So net movement of water out of the cell, that's true. Is the cell going to burst? Well, if water are leaving the cell, it will shrink and not burst, right? The cell will only burst if it's an animal cell. It's not bursting, it's shrinking. The concentration is higher outside compared to the inside. That's true. That's why water is leaving. Okay, so this is slide 47. Answer are A, C, E. Okay, and then we keep on moving forward. Skip, 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 skip. These are all the stuff that I was writing the note for, right? Like feel free to read the slides yourself and have a better understanding. That's the human egg right here doing the penocytosis. Okay. Okay, so here. What slide is this? This is slide 60. Right, 60. Right. If you don't have the PowerPoint slides, just write down 60 and then write down the answer so you can check later. Which of the following, sorry, I really gotta edit these slides, can cross the cell membrane easiest? Okay, so let's take a look at the first option. Small charged molecules. If it's charged, they can cross easily. That's why they need the channel protein, right? So that's not easy. I mean, it can cross, but the question says easiest. Okay. Large hydrophobic. Hydrophobic is good, but large is not easy, right? If it's too large, it again needs help, right? You might need to do phagocytosis. You might need to do carrier protein. It's not the easiest. Small and hydrophobic. That's the best one. That's the easiest to cross. They can do it by themselves. Okay. Hydrophilic is not easy. Right? You need a channel protein for that because the membrane is mostly hydrophobic, right? And then bacterium, not easy. You gotta eat them. You gotta do phagocytosis. Okay. All right, this one. I'll read it to you. Let me know what you think. A patient has had a serious accident and lost a lot of blood. To replenish body fluids, distilled water, which is just, you can think of it as like pure water, equal to the volume of blood lost, is transferred directly into one of his veins. What will be the most probable result of this transfusion? What do you think? Is this going to save him? Or is it going to make him worse? Is it going to make
Even worse.
why is that? Yeah, a few people are saying it's worse. Patricia, you say worse. What, you want to justify your reason?
Yeah, I'll try.
Sure, please.
I think it's worse because he's lost a lot of blood and we're pumping water in his vein opposed to blood. So he has like his white blood cells are high
Okay.
and his red blood cells are decreasing.
Okay.
I
I think you're on the right track. Now, we don't really know what happens to his red cells or white cells, right, from the question, right? I mean, if you bleed out,
Yes.
you are losing both, okay? But I think you're on the right track. Anyone else want to add to that, maybe?
Like, it's because distilled water is hypotonic compared to the blood.
That's right. That's the key thing we have to say, right?
So it will
Yeah.
cause the water to rush into the red blood cell and
That's right.
make it to burst.
Exactly. That's the key thing I want to hear if this question shows up on like the test or something, right? Hypotonic, the distilled water is gonna basically dilute the blood. I think someone else said that earlier, right? Like who is RJ, right? Yeah, it's diluting the blood. It's gonna create a hypotonic environment for the red cells. The red cells will swell up. They might burst, right? So it's gonna make him worse because he already lost a lot of blood, a lot of red cells. He's probably a little bit anemic. We call these hemorrhagic anemia, right? Anemia caused by blood loss. And if he continues to lose more red cells, then he would have trouble basically transporting oxygen. So this is not the right thing to do. Okay. Good. Now, some of you told me last time, I forgot if it was your class or the other class, but like, the people were complaining, well, not complaining, but okay, well, yeah, complaining. People were complaining that some of the questions on the quiz were phrased strangely, and that, it's not like the way that we talk in class, right? And that made it difficult to do the question. And I said, I'm not the only one who's making up these questions, right? That was the explanation. There are other profs teaching the course, but this is one of them, right? This is one of those questions where you look at it and you're like, oh my goodness, so many words, right? What are we supposed to do? So let's take a look. I'll read it to you. 5 bags constructed from a semi-permeable membrane. So semi-permeable means some things can come in or leave, others cannot. Okay, and it says here it's impermeable to sucrose. So it does not allow sugar to pass through. That's basically the first sentence. And then you fill them up with various concentration of sucrose and then place them in separate beakers containing initial concentration of 0.6 m sucrose solution. Now you look at this and you're like, 0.6 m, what does that even mean? it doesn't really matter. Okay, M is the measurement of concentration. The key thing to read here is that there is an initial concentration of some number. All right, so we can put 0.6 on the outside. And then we're gonna have a semi-permeable bag here. And it says which line represents the bag that contains the solution isotonic to the 0.6 molar solution at the beginning of the experiment. So if it's isotonic, what number should I put in the inside? Should I put a number that's higher than 0.6, equal to 0.6, or less than 0.6? Justine says equal. That's correct. That's what isotonic means, right? Same, right? So what's going to happen? Well, if it's the same, then you would have water coming in and then you would have water coming out. And the rate at which they come in is going to equal to the rate at which they leave. So at 10 minutes interval, the bag were weighted and the percent change in mass of each bag was graphed as shown below. So which graph represent the change in the mass? Yeah, go ahead, Patricia. I see that you unmute. Go for it.
I'll try. I think C.
C, absolutely, right? Okay, that's the one. It doesn't change because it's isotonic, right? So never mind all these mumbo jumbo, right? The key thing here really is just the fact that it's isotonic means that there's no change. Nothing's like things are coming in and things are leaving, but they cancel each other, right? Now this top curve here, that would represent, hopefully you agree with me, hypotonic, okay? Because the volume, the water is coming in, making it heavier, so it goes up, but then it doesn't go up forever, right? Like after a while, it's gonna stop changing. Okay, the bag is gonna fill up, and then like the bag's not gonna burst, right? This is a bag, not a cell, right? So it swells up, and eventually, water will stop coming in because the concentration has been equilibrated, right? Has been like made the same inside and outside, right? Okay, so that's why it's gonna be flat. It's not like B, right? B implies it's gonna keep on going up forever, right? Which is not true. Same thing for E, it would be for hypertonic, right? Where the water starts to leave and then it shrinks and then eventually it plateaus out, it flats out. Okay, are we clear on this? Right? Don't just choose C on the test. Read the question carefully, see what kind of solution it is. All right, now this question, many people get it wrong the first time they do it. I want you to try it, and then I'll explain, and then it will help you do one of the lab questions. Okay, read it. I'll launch the poll in a second. All right, here we go. Please choose something. You have a 33% chance of guessing it right if you're choosing it in random. Anyone else? Okay, I'm missing 3 people. Who are you? 2 people now. Choose something. All right, I'll stop bugging you guys. Okay, here are the results. Interesting, right? Like we have almost like an equal split. Okay, so let's talk about this. Now, a lot of you read this and you're like, oh, hypotonic. The big O means water is gonna come in. It will swell and burst, and that's why you chose A, right? Those of you who chose A, I think that's what you were thinking. Am I correct? Probably, okay. But But the key here is freezing and thawing. Freezing and thawing. Now, I asked this question to my morning class. I was shocked to hear what they had to say. Right? So I'm gonna try to ask again. Tell me what happens if you put fill up a plastic bottle with water, you put it in the freezer, okay, and then you take it out like after 3-4 hours. Okay, obviously the water is frozen, right? But what else happens to the bottle? Do you guys know?
It expands.
Yes, it expands. Thank you. Does the rest of you know that? Okay. People were telling me it will shrink, it will, you know, do other weird things. Like, I don't know. It feels like nobody has freeze water bottle before. Okay. But it expands, right? Are we okay with that? So, if you have a cell and you freeze it, the cell will also expand, right? And the ice crystals from the freezing will actually puncture, right? It will like poke holes in the cell membrane. Okay, ice crystals are really sharp, right? So after you freeze the cell and then thaw it, the cell membrane is full of holes. And then when you put it into a solution, it actually doesn't matter what kind of solution it is. Hypotonic, hypertonic, isotonic, it doesn't matter. The whole thing is full of holes, right? The whole cell membrane is full of holes. So water will just go in and out without any kind of regulation, right? So the cell is not going to change size. Okay, does it make sense? Like stuff is just going to leak all over the place out, and then things are going to go back in, no change. Does that make sense?
Yes.
Yes.
Yeah. Now, usually at this point, someone in the class is going to ask me a question related to reproduction. Okay, does anybody have a question like that? Now is a good time to ask.
Well, I have a, but not about reproduction
Okay, sure.
necessarily.
Okay, go for it.
Okay.
Yeah, it's okay. Yeah. I'll tell you
Wow.
what the question is after. It'll make sense. I know, right? It seems like it's so weird, right?
Okay.
Yeah.
So my question is basically, okay, after you freeze it and then like how do you you can't necessarily know if it's gonna, like, yes, it's gonna expand, but
Yeah.
what if it doesn't burst?
Oh, so it's not about bursting, right?
Or
This is about like the ice crystal, like ice crystal, like when water freezes, right? Like it basically forms these kind of like snowflake structures, right? And those crystals are what pierce the cell membrane, right? Like basically poke holes into the cell membrane, right? And that's what we know, right? Now, one way to think about this is if you ever thaw frozen vegetables, like if you buy like, you know, pre-cut frozen whatever, like broccoli or something, and then you just thaw it in a bowl, the water that comes afterwards, it's like green, right? So like that's the chlorophyll leaking from the cells, right? Like if you soak a broccoli in like a regular broccoli at room temperature, you soak it in water, the chlorophyll is not going to leak out because the cell membrane is intact, right? But if you put it in a bowl, like put frozen one in a bowl, right? As it's thawing, all the chlorophyll comes out because of, you know, the damage from the ice crystal. Does that answer your question?
yeah, I guess so. So what you're saying basically is like the ice crystals will poke holes because
That's right.
it's frozen, so the membrane will be pierced.
That's right, because the water is expanding and then it's like all these sharp crystals coming out and then basically leaves the thing full of holes. Okay.
Right.
So whatever's
Okay,
inside
gotcha.
will leak out and then things will go in without regulation and so no change in the size, right? Okay.
Okay.
All right. And now the reproductive question. Now, most people don't do this, okay? But like, you know, celebrities, they like to because they have too much money, right? So like, you know, they can do other things that ordinary people like us don't do, right? For example, they could choose to freeze their eggs, right? For later on in life when they decide to have a baby, Okay, so like they might not want to have a baby early on, and I mean not just celebrities, right? Like women have that choice if you're able to afford it, right? To freeze the egg for later use, right? If you want to delay, having a child later on in your 40s, for example. So your eggs are still fresh, right? Sperm banks, right? Donors will donate their sperm and then they would freeze it, and then you can thaw it and then use it for in vitro fertilization, for example, or you can donate it to other people. So the question people sometimes ask me is, how come it works? Like if you freeze the egg or sperm and then thaw it later, according to what we just learned, the egg is destroyed by the ice crystal, right? And the sperm is destroyed by the ice crystal, right? So what's going on, right? That's the question. It's actually quite simple, okay? They actually add a little bit of antifreeze to the solution that the egg and the sperms are kept in. Kind of like your windshield fluid, right? But these are like non-toxic version antifreeze, and it basically prevents the crystal from forming. Okay, so you can cool the temperature without forming the crystals. You don't pierce the cell membrane. The integrity of the egg and sperms are maintained. Okay, and now we know. All right, that is it for the lecture. Now I'm gonna go ahead and talk about the lab. All right, how many of you have done the lab yet? Like, have you guys tried some of the questions? Did you watch the video?
Yes.
Yes. Okay, some of you did. Yeah. Okay, good. Okay, well, a few people did anyways. So again, above all else, you must watch the video. Okay, that will give you an idea of what the experiment is. I'm actually, doing the in-person class tomorrow. My in-person class will be doing the experiment like physically, but since you guys chose the online version, you will have to watch someone do it, which is totally fine. Last class, I told you the purpose of the experiment, right? So I'll say it again in case you missed it. You might not be able to write down everything I'm telling you right now, but you can watch the recording after, right, to catch it. I'll be explaining the questions one by one so that you know exactly what I'm looking for. All right. So first question, what is the purpose of the experiment? The purpose of the experiment is to examine the effects of temperature on beetroot membrane permeability. That's all you have to write. Okay, you don't even have to change my word. You can write it exactly the way that I said it, and I'll give you 2 marks. All right, you're welcome. Right? Just write that down. Don't even change it.
Excuse me, sir. What if this is the way I put for my own question one? I said to investigate the effect of temperatures on permeability of cell surface membrane of beetroots.
Yeah, sounds great. Okay, it's essentially what I said, right? Okay, that's great. I mean, you don't have to say exactly what I said. You can use your own words, but I'm saying to make it easy, yeah, you can just copy what I said. All right, and then I will give you 2 marks. All right, next part. Write the hypothesis for this experiment, right? So the hypothesis should be something like this, okay? Increasing temperature will blank, 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 thus membrane permeability. Okay, you either tell me that you think temperature will increase the permeability, membrane permeability, will not affect the membrane permeability, or will decrease the membrane permeability. Okay, just choose one of those, put in the blank, fill in the blanks, and you're done. Okay? You should be taking notes, all right? This is 10%. It's gonna boost your grade tremendously if you do well. Next, state the dependent and the independent variable. The dependent variable is the one you are observing in the experiment. So after you watch the demo, what was the lady observing? What was she measuring in the experiment? And then the independent variable is the one that's being changed. Okay, there's only one thing that was changed in the experiment. Okay, so all you have to do for this one is type dependent variable, and then whatever you think it is, and then independent variable. Okay, and then da 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 da. That's all. 1 mark for each. Got it?
Yep.
Okay. Now this one people always messed up. Let me try to explain to you what I want. Okay. The question asks, why is beetroot chosen for this experiment? Example, why not use carrots or tomatoes? So a lot of people, they put, because beetroot has a bright red color. Okay. Be that as it may, that doesn't explain why it is a good choice for this experiment, right? I mean,
Yeah.
carrots are orange and tomatoes are red. They too have colors, right? So why are we not using those? Okay, why specifically to choose beetroot? Okay, so when you answer this question, I want you to think about if you were doing what the lady is doing in the video, but with carrots, would you still be able to achieve the same outcome? Or are you going to run into some kind of difficulties? Okay, same thing with tomatoes, right? Are you going to be able to do the exact same experiment? Or is there going to be some part that is not going to really work out? So you want to tell me what is it that makes carrot and tomatoes not so suitable for this experiment? Okay, sure, you can say the beetroot has a bright red pigment, okay? But keep in mind, carrot and tomatoes also have those pigments. So what's so special about this pigment, right, compared to the other ones? You don't have to look it up online. Okay, in fact, you won't find answers online. Okay, you just think about it logically, and see if you can find the answer. Is that clear? Do you have follow-up questions? No follow-up questions. Great. Like 2 marks, okay? Don't write too much. Just like a couple sentences, 2 or 3 sentences, you're done. Okay. Be clear, be concise, and be correct. And use your own words. List 4 safety precautions. All right. So this is what, again, I'm telling you what not to do, right? So that you don't lose marks. In the past, people like to put like, wear gloves, wear goggles, wear lab coat, and wear, I don't know, something else, okay? But they are all the same thing, okay? They can all be summarized into wear protective personal equipment, right? PPE, right? Okay, so that's just one point, right? I want you to think about other things. Does anybody have any safety precautions that they want to share with the class? Remember, whatever you say
Yes.
here, other people might use it. Okay, go ahead.
Yes.
Yeah.
So the first one I said, dispose of the beetroot solution in a safety manner.
Dispose the what solution? I'm sorry.
The beetroot solution.
Okay, yeah. Yeah, sure. But I mean, like, beetroot is not that dangerous, right? Like we can eat the beetroot, right? So like that's not, I wouldn't say that's a really high priority safety precaution. Okay, but that's good. Justine said label the tubes. Again, labeling the tubes, we're not dealing with like, very concentrated acid or something like that, right? So labeling the tube is more like for procedural reason. It's not really for safety, right? Yeah, for the temperature. Okay. Can you elaborate a little bit, Justine, please? You might be onto something.
Like for example, like in the picture, right? You can't really know like the tube. Like you can't really know how hot like the tube is. So like label them so like you won't get like burned or
yeah, so
like,
that's
yeah.
good. Yeah, exactly, right? So take precautions while handling hot glasswares, right? That could be a precaution, right? Does that make sense? Okay, so don't just use one word, like elaborate a little bit, explain a little bit, right? Okay, is that clear what I want? Maybe one more. Anyone else have anything else that they want to?
How about for watch your hands only after the experiment is complete.
Yeah, but you're wearing gloves, right? Aren't you? Yeah, I mean, sure, you can wash
What does
it.
it mean?
You
Yeah.
can say as a precaution that, let's say if something, one of the solutions or something like that was spilled, make sure that it is properly like cleaned up
Yeah,
before
sure, right? So clean up any
washing.
spills, right? So people don't like slip on it or whatever, right? Okay, that's good. All right. So think about it. Okay? And you're using knives, guys, right? Supposedly. So, you know, precaution for using knives. Think about that. All right, moving on. These are procedural stuff now. Test tubes were, I really got to fix up the grammars of these things. I'm so sorry. Test tubes were labeled 20 degrees, 40 degrees, 60 degrees, and 80 degrees. Which test tube is considered as the control of the experiment? Okay, so this is how you should answer it. Okay, the control of the experiment is the blank test tube. Okay, is it the 20? Is it the 40? Is it the 60? Is it the 80? Those are the only choices. Okay, don't make up anything else. All right, I got people who put like bizarre things as the control because they look it up on Google. They didn't watch the video, right? People would say the control of the experiment is the test tube with alcohol, and I'm like, what alcohol? Like, there's no alcohol in the experiment. It's just water. Like, have you been drinking alcohol, right? Like, so it should just be the 20, the 40, the 60, or the 80. Okay, that's all.
Like, I don't know if I'm right.
Yeah, but don't tell the whole class the answer, right? Okay. Keep it to yourself. All right.
I already turned my assignment in.
That's good. That's very good. Okay. Now, the control for those of you who are struggling, right, is the one that has not been modified, right? The one that has not been changed, right? Okay. Think about the example we did in class, right? Okay. The people receiving vaccine and people who are not receiving vaccine, right? The groups that's not receiving it, that's the control, right? So which one is the control? That's the first part. And then the second part is explain what is the significance of having the control. Okay, then you should follow up. The purpose of including a control in the experiment is, and then you give me the reason. Okay, like, why do we have to have two groups? Why can't we just give everybody the vaccines and see if it works or not? Why do we need to have a control group that's not receiving anything? Okay, what's the purpose? What's the significance of having the control? So make sure you answer those two things. Is test tube like not pearls? Interesting. I did not know that. And then you would have 2 marks. Okay, just 3 or 4 sentences. All right, keep it short. All right, next one. What needs to be taken into consideration when cutting the beetroots? This is another one where I don't know if it's the way I phrased the question or if people are just like misinterpreting it. I am not asking about, be careful not to cut your fingers kind of thing. Those are safety precautions, right? When I say what needs to be taken into consideration when cutting is how you are cutting the beetroot, okay? So I'll give you an example. Like, you want them to be the same size, right? And then you have to explain why that is important. Okay, like what happens if you don't keep it in the same size? Like, what if I have some big chunks and some small chunks? How is that going to affect the result? Okay, so besides size, is there anything else we need to take into consideration? Okay, so size is only one of them, right? I think about other things that you have to consider when you're cutting the beetroots. And more importantly, you explain to me, if I don't do that, how will that affect the accuracy of the experiment? Okay? Again, you might not get everything I'm saying right now. Just watch the recordings afterwards again and listen to it. Hopefully it'll make more sense. You are welcome to email me and ask me for clarification. Okay, I won't tell you the answer, and I won't look at your answer and tell you if it's okay or not. Okay, so don't send me your answers. But if you want me to clarify a question, you're welcome to do that. Okay, just say like, you know, can you explain this one again, like in more detail? I can do that for you.
How many are you looking for? How many?
So I gave you size, probably one more, right? Right, 2 marks.
Okay.
Yeah.
Okay.
Why is it necessary to rinse the cut beetroots afterwards? Okay, so you see the lady after she cuts it, she washed them and then pat them dry. So tell me why she needed to do that? What if she doesn't do that? How is that going to affect the results? Okay. Next one is another challenging one for some people. Why is it important to leave the beetroots in the test tube submerged in the respective water bath for 20 minutes? That is, why not longer or shorter? Okay, so she did not explain why in the video. Okay, she doesn't say why 20 minutes, but I want you to think about why. Okay, what if you do it for 5 minutes? How will that affect the results? Are you still going to be able to get the same result, or is it going to be inaccurate? And if you think it's going to be inaccurate, why? Okay, what if you do it for longer? Right? Why not do it for 30 minutes, an hour? Right? How's that going to affect your result? That's what the question is asking. Okay. Any follow-up question for this? Again, I don't think you can find anything on the internet. Just use your just try to use your intuition. Okay. And don't I know some of you probably have, like, you might know another person in the class, you guys might have a chat group or something. Really try to do the question yourself. All right, because what ends up happening is, like, someone in the group came up with an idea and everybody decided that to go with the same explanation, and sometimes it turns out to be wrong. Okay, these are things that you can think about yourself. Okay, what happens if it's too short? What happens if it's too long? Moving on. Describe the correlation. Correlation is like a relationship, right? Between the temperature and the color intensity. So looking at this test tube right here, the rack, okay, we have the various temperatures, right? What happens as the temperature increases? So this is how you answer. Part A, as the temperature increases, the color intensity, and then you finish the sentence. Like, what happens to the color intensity? I think it's pretty obvious what's happening. Okay. Next. The answer you gave in part A is an example of a qualitative analysis. Explain what this means. Again, many people just copy this into Google and search qualitative analysis and then paste the answer here. That's not what I'm asking you to do. We learned about qualitative analysis in lecture 1, okay? Remember, when we do observations, it could be qualitative or quantitative, right? That's what this is asking. What is qualitative, right? That's all you have to tell me. Qualitative observation, qualitative analysis involves using, right? Look at your notes. Next, in this experiment, how was the result measured quantitatively? If you've watched the video, not only does she judge the color intensity, she also has an instrument that measures the intensity, right? Okay, so whenever you measure things, right, that's quantitative, right? So you want to tell me how she did it, right? Does describe the relationship between absorbance measured by spectrophotometer. I think she calls it something else in the video. I haven't watched it like in a long time. I think she called it like a colorimeter. Okay. Some people write like color emitter. I don't think that's what it's called. Okay, it's called colorimeter. It's a meter that measures color. Okay, colorimeter. Right. So just even though the captions might say something else, but remember the captions are auto-generated on YouTube, right? So it might not be accurate. Okay, colorimeter. I think that's what she's calling it. Okay, so describe the relationship between absorbance and measured by the colorimeter and the color intensity. So let me just go through this with you. Yeah, a lot of people don't know this, but if I have a red circle, okay, if I have a red circle, Okay, and light shines on it. Okay? What color of light is reflected by the red circle? Like your eyes are here. This is your eyes. Okay, what color of light is reflected off a red surface so that you perceive it as red?
Is it like inverted into like the opposite?
Maybe. Anyone else? If something is red, what color is bouncing off its surface? That's my question.
Red.
That's right. There is no, it's not a trick question, okay? It's red because it absorbs every other color except for red. Okay? Do you understand that? Right. Now, somebody say yellow. Now, yellow is a different situation. Yeah, yellow is do you know there are 3 primary colors for light? Okay. What are they? My morning class also struggled with this a little bit. There are 3 main colors that makes up all the color in the world. What are the 3 main colors? That's right. Red, green, blue, right? Okay. RBG, right? Red, green, blue. And yellow, if something is yellow, that means it is reflecting, believe it or not, blue light. Nope, lying. It is reflecting green light and red light. Did you know that? So it absorbs everything except for green and red. And when those bounces off and enters your eyes, it becomes yellow, right? Look, if you don't believe me, because my morning class absolutely did not believe me. They were like, no way, right? Yellow plus blue gives you green and stuff like that, right? Those are for like painting color, which is different than light color. I'll show you. Look, this is a blue circle. And what am I doing here? More colors, okay. So that's a blue circle. All right, now I have 3 colors, red, green, and blue, right? I'm gonna delete the blue. I'm gonna make blue 0. So only red and green shows up. See, we got yellow, right? Okay, so it's yellow because it absorbs all the blue and then it reflects the red and green into your eyes and you see it as yellow. Okay, if you have one of those old TV at home, the one with the big, cathode ray tube at the back, right? Like when you wipe the screen with a piece of red cloth, you can see the individual pixels. It's either a red, blue, or green pixel. Okay. Anyway, that's your physics of the day. So back to here. If something is red, it absorbs everything except for red light, which means, right? What we can do is we have the test tube, and the test tube is different intensity of red color, right? Depending on how much beetroot juice pigment has leaked out. So the colorimeter basically creates a green light beam, green light, and then there is a detector that detects how much green light passes through. Okay, the more red it is, the more green light is absorbed, right? So we call that absorbance. Absorbance is how much light is being absorbed. Example, if it's really red, then it might have like 80% absorbance. 80% of the light that is sent through the solution has been absorbed. Only a little bit went through. If the color is really light, then you would have a lower absorbance, right? If it's not really red, if it's like, just, very light kind of red, it's almost pink, right? Then you would have lower absorbance, right? Because it's not picking up all the green lights. Okay, so hopefully that explains to you the relationship between absorbance and color intensity. Okay, so all you have to do is tell me high absorbance means and then low absorbance means something else. Any follow-up questions? Again, use your own words. Okay, I cannot stress the importance of like using your own words and refrain from looking it up online, because it sounds really weird and out of place when you use something online. It would look very different from the rest of your report, and that's how I know when you have copy and answer from online. Okay, next one. You have to use the data here to create a graph. I've created a YouTube video that shows you how to do this on Google Sheets. I'll post that after class. It's very easy. It will take you like 10 minutes, and then you put the graph here. It's 4 marks. Okay, so make sure you do it properly. Okay, I need to have the right labels. I need to have the right title and stuff like that. Okay, so just copy and paste your graph directly into here. Okay, do not give me a link and tell me to go download it somewhere else, right? A lot of people do that. I don't know why. Just copy the graph right here into the document. Okay, next one. At what temperature does the absorbance peak? So I want to clarify this as well. If we have a graph here, okay, and let's say the graph looks like this, okay? Where is the peak? Is it A, B, C, or D? Where is the peak?
C.
Okay, someone says C. Okay, Patricia says C. Jay says C. Anyone think it's not C? Okay, Blessing say D. Anyone else? Come on, tell me what you think. It's like so quiet, you guys. Right, so like those are the common choices, right? I'm glad no one chose A and B. At least you didn't say it out loud, right? You know, they say C or D, but the answer is C. The peak is at C. Okay, because after C, it's like not really changing much, right? Sure, it might be going up slightly, but you know, it's not changing drastically afterwards anymore, right? So like that's the peak, right? It's no longer changing significantly afterwards. So remember that when you answer this question. Part 2, do you expect the temperature where the absorbance peaks would be the same for membranes of other plant cells? Like what if we do the experiment with cabbage, right? And then we measure the absorbance and stuff like that. Do you expect them to peak at the same temperature? All right. So this has to do with membrane composition. Yeah, think about the things that we discussed in lecture 4 with the you know, with the saturated and saturated fat. That will give you some insight. Okay. So that is 2 marks here. Make sure you explain properly. Provide 2 reasons that explain how temperature affects membrane permeability. That is also 2 marks. The lady in the video actually, she gave 2 reasons towards the end. Okay, it's very clear what she said. Okay, just copy what she said, paraphrase it, and then you're almost done. What is the conclusion of your experiment? Right. So looking at the data, what do you think? Do you think temperature affects the intensity? Right. Does it agree with your hypothesis? If so, you would accept it. If not, you will reject it. I'm not grading you based on whether you got your hypothesis right or wrong. Okay, I'm grading you based on whether your conclusion is correct or not, and then you tell me whether you accept or reject your hypothesis. That's it. Now, number 4, you used to be that, you know, people always get it wrong. But hopefully you will get it right after I explain to you the freezing and thawing thing. Let me read it to you. A piece of frozen beetroot is placed in the test tube of tap water at room temperature. The absorbance recorded after 20 minutes is 84. Provide an explanation for this. So let's see. Absorbance is 84. And if I go up to here, 84 corresponds to, you know, I would say like maybe around 50 degrees, right? So why is it that the beetroot is frozen, but then the absorbance corresponds to a much higher temperature, right? Why is this happening? That's the question, right? So hopefully, using what I told you, you will be able to answer that. Okay, so the lab is due on Thursday. you know, before midnight, just submit it on eCentennial. And remember to use the proper format, right? So I can mark it, I can open it and mark it. Yeah, that's it. Any last minute questions before we say goodbye? No class on Thursday, huh? You just work on the lab. Yes, BB?
When is our...
Yes.
Sorry.
Yeah, go ahead.
Okay, so the question that I had to ask was, we are just basically downloading this template and putting our answers inside.
That's right. Yeah, don't change anything. Just type the answer in the box, okay? No need to make a title page and all that nonsense, okay? Just type the answer in the box and submit it. That's all.
Thank you.
Okay, and Tahib, you have another question?
Yes, sir. I was trying to ask, when is our next quiz?
Quiz is on Tuesday, okay?
Next week, Tuesday.
Yeah, right after the long weekend. We will have a class on Tuesday, so please plan to attend. I will post the information about the test after class. Okay.
Thanks.
Yeah. Anyone else before we sign off here?